Hi everyone, I'm Michelle Cooley. I'm a rising junior majoring in biology. Something about me is that after graduating from UNC, I hope to attend medical school to study pediatric infectious diseases and eventually practice in Spanish-speaking countries. I'd like to become a chemistry mentor because I've discovered through teaching my peers, I learn different ways to approach a problem and the material in general, so I also get a better understanding of the chemistry topics we're talking about. Also, through my different classes at UNC, I've heard of tons of different companies and individuals offering chemistry tutoring, but typically their rates are so high that it's unaffordable for many students and their families. So I think being a mentor is a great way to help other students in getting the in-class assistance and out-of-class assistance that they need or want, but that they wouldn't be able to otherwise afford if it were from a private tutor. So today I was gonna help explain a problem regarding the HNMR signals for a certain structure. So HNMR, or also called proton nuclear magnetic resonance, is a form of spectroscopy that helps to give information about the structure of a given molecule. So this question is asking how many signals will this molecule produce on a spectra. So the number of signals is the same thing as asking about the unique chemical environments, or in other words, how many different types of protons are there on a molecule. So the best way to approach that problem is just start grouping protons based on which ones are identical, or technically called equivalent protons. So starting at the left side of the molecule, all protons within a methyl group are known as equivalent in that they're in the exact same chemical environment. So these three protons will all produce one signal together, as will the protons within this methyl group. All three will produce one signal together. However, when you look at the two methyl groups, they are actually also chemically equivalent. So these 6 protons will all produce one signal together. So we have one signal right here, and then we hit this hydrogen right here. Since there are no other hydrogens like that within the molecule with these types of connectivities to 2 methyl groups and then a CH2, this will produce a second signal within the molecule. Then jumping over to the right side, there's another methyl group. So the 3 hydrogens will also produce all one signal together, and since there aren't any other hydrogens or methyl groups connected right around here to the exact same carbon, then this will produce a third signal within the molecule. Now this hydrogen attached to the oxygen, it's the only hydrogen attached to this oxygen, and so that will produce a fourth signal, as will this hydrogen that's attached to the carbon. Since there are no other hydrogens identically attached to this carbon, then that will represent a fifth signal, because there aren't any other hydrogens with these connectivities throughout the rest of the molecule. Now the tricky part comes down to these 2 hydrogens in the middle. At a first glance, they look like they're equivalent, and thus they're going to produce one last 6 signal together in the molecule. However, when you look actually at it, these 2 hydrogens are known as diastereotopic, in which case they're chemically inequivalent, so they actually produce 2 different signals. So a quick way to figure out if protons are diastereotopic is you do the substitution. So substitution is when you take one of the hydrogens and you actually substitute it for a deuterium, and you check to see if the stereocenter thus has an R or an S configuration. In this case, it goes to an R right here if you substitute the deuterium. And then you actually switch this side, leaving that as a hydrogen, and this becomes a deuterium, in which case it goes to an S. Because one stereocenter is changing its configuration while all the others stay the same, these 2 hydrogens are going to be diastereotopic, in which case this hydrogen, or that one, will make a 6th signal, and then the other one will make the 7th signal. So in total, the question is asking how many signals will this molecule produce on a spectra. So the predicted number is going to be 7 different signals on the HNMR spectra. So that's the final answer, and that's how you can figure out the number of signals produced